But Minnie, how many more seminars do we have today?
Thanks for joining, Marilla. You too.
That, that these speakers are too good to miss.
Yeah, just
Have a better help. How are you? Good, how are you? I'm doing well. Sure.
I think we should get started. Good afternoon, everyone. I hope you all had a cup of coffee. There's cookies and coffee on site. So this is really a treat to have two eminent TB researchers amidst our midst today and giving us seminars. So thank you both again. And so the afternoon seminar is by Dr. Gerhard Walzl from the University of Stellenbosch. Currently he is Head Division of Molecular Biology and Human Genetics, Department of Biomedical Sciences, Faculty of Medicine and Health Sciences at Stellenbosch University. So as Bruno mentioned earlier, Gerhard is really a pioneer in the field of TB biomarkers and has done some real seminal work in the area. I think most of us here know of his paper, the four-gene bio-risk or risk progression marker of TB disease. This is a paper he published with Salman a few years ago, and it's really has been a major seminal contribution to the field of biomarkers in TB. He continues to work in the field using both proteomics and gene expression as platforms to look at TB biomarkers, both of progression to disease and treatment failure or relapse. Gerhard has, again, large number of NIH grants in collaboration with many investigators here in the US. He also has several what's called the European EDC-TP grants, and also funded by the Bill and Melinda Gates Foundation. So he runs a really well-funded lab to look at, as I said, biomarkers and diagnostics in TB. Gerhard has close to 300 papers, again, all in very high-profile journals, really making a significant contribution to TB research. So it's really a privilege to have you here today and talk to us about biomarkers.
Thank you, Padmini. Thank you, Jerry, for the invitation. So, in a way, Bruno did me a lot of favors by his talk. Because we use a slightly different approach to it. And I think it is a complementary approach to yours. So in a way, we embrace the noise, although we are very careful to characterize our participants as well as possible. I think a biomarker or a biomarker signature has to be able to shout over the noise, because patient cohorts are noisy. And I'll try to show you how we try to do this. Okay, none of these seem to work.
We have help right
That
there.
is not my only slide. There we go.
Thank you.
Thanks. So a lot of you would have seen this slide. This is published by the Gates Foundation, and it basically shows that we cure about 50% of TB cases. Because at every step of the way, from getting to a test, to being tested, to being notified, to starting treatment, to completing treatment, we lose patients. So 50% cure rate really is not great, although the cure rates that the WHO publishes really use the people started on treatment as the reference. And we know that certainly in South Africa, about 76% of people who start treatment are cured. That's actually quite shocking. And the international average is about 85%, which is equally shocking. So how can biomarkers help here? Bruno wants to use it to understand biology. Yes, absolutely. We need to do that. But what we are also trying to do in parallel is to find diagnostic markers that can replace the rather cumbersome diagnostic process that patients currently have to go through, where we know that people who deliver a positive sputum, about a quarter of them never go on treatment. And that is because it's not a one-stop service. The sample has to travel to a central lab. The result has to travel back. The patient has to come back for the result and loses income on that day. So a quarter of people who should be treated, where we have the information that they have active TB, are never treated. Prevention, of course. Padmini mentioned the biomarker work that we've done to try and move towards targeted prevention, because there are just way too many people in our country, for instance, about 600,000 new TB cases each year. They all have, 5 to 10 close contacts in their houses. There's no way we can give them all preventative treatment. So can we find the people who need prevention most? And then treatment response. When is treatment enough? We overtreat everyone to minimize the risk for relapse. And we do 6 months, and we overtreat 85% of people, because we know that 85% of people can get away with 4 months of treatment. We just don't know which 85%. And that would help us in evaluation of new drugs and regimens as well. So this is how I understand TB. Maybe wrong. But for me, the important line here is this line. Oh, yeah, that's very small. This line here, the limit of detection of our microbiological tests. I don't think we desperately need anything in place of the microbiological tests, although, like I said, it is a cumbersome process. Smear is useless. Well, smear 60% sensitive, can be done on site. Expert is great, but it's not that widely available, etc. But the problem is this line, the limit of detection. Anything under that limit of detection poses a problem. If we look at subclinical TB, can you see the pointer at all when I move?
Yeah.
And move it slowly, probably. So this is subclinical TB, and this is what I think we detected with our signature markers in the adolescent cohort study and the GC6 study, is people with negative sputum or with occasional positive sputums that go away again. So this is an area where I think we have opportunities and important opportunities. And then again, after initiation of treatment, after about 2 months, you fly below the radar again, you have no clue when to stop. So we treat for 4 more months. We know that if we stop early, more people relapse. So this is another area where I think that host-based biomarkers can help, until the day when technology catches up and can detect very low levels of microbacterial proteins or the whole organism. That is currently not possible. So I regard host-based biomarkers in a way as water treading until microbiology catches up. Where's David Maland? He needs to find that test. And things like urinary lamb maybe, if we can get more sensitive urea lamb tests. So extrapulmonary TB is another problem that where we need a better test and where host biomarkers may help. So the thing with host-based biomarkers is that the immune system has very sensitive ways of recognizing trouble. And if we can test markers that are released by the immune system, they may help us to find a looming risk. So here we show two different studies, the adolescent cohort study that looked at adolescents and their progression to active TB, and a risk signature was developed on whole blood RNA transcripts, and that risk rose as people get closer to their TB event. We used the same signature in a treatment study, a different treatment study, that indeed showed a very high expression of that risk at baseline, and that gradually goes away to control levels. And this symmetry suggests to me that we have some hope of using a limited set of biomarkers for many different parts of the host pathogen relationship. So we started with antigen-specific responses, lymphocyte or whole blood stimulations, measured cytokines, cytokine expression, activation markers. The problem with that is it doesn't lend itself to point-of-care tests, which is where we will need such tests, because you need a stimulation period. Serology lies between the antigen-specific and the ex vivo markers, and is a attractive alternative, and Marilla has worked on this in many very interesting large studies. So we have moved on to cytokines, mRNA, microRNAs, metabolites, proteomics, because all of those could potentially be developed into point-of-care tests. And by using more than a single marker, using 3, 4, 5 markers, you can possibly overcome the lack of specificity of a single marker. Because a particular disease might be associated with a fingerprint, with a pattern of cytokine abnormalities. If you look at some of the other stuff out there, like symptoms, X-ray, CRP, kind of using these as benchmarks, we see that they're not fantastic. They either have good sensitivity or good specificity, but never both. CRP on its own is probably the closest we can get to a reasonable test, 77% sensitive and 74% specific. That's not too bad. But as you see, even expert only has a 68% sensitivity, but a very, very high specificity. So the WHO target product profiles, based on this and a wish list, suggest that we need, for a non-sputum triage test, at least 90% sensitivity and 70% specificity. And we are aiming for that. Well, we're aiming higher, but I'm not sure we're going to get there with host biomarkers as things currently stand. So what we did is, to shout over the noise, we did multiple rounds of investigations, improvement of biomarkers. We started with a study of about 1,000 people across different African sites, and we measured a whole lot of analytes, and we came up with 3 CRP, serum amyloid protein A, and IP10 that differentiated on their own, but even much better so in combination, people with symptomatic tuberculosis from those with other causes for their symptoms. And this was in peripheral healthcare clinics, not in hospitals. And we had 3 rounds of EDCDP funding for that. And the last one actually, it also includes a FEND project. That's an NIH-funded project, similar to the one that Jerry leads. And we have done head-to-head comparison between our 3 host protein fingerstick test and the Gene Expert host response cartridge. So you can see here that we started off with pretty primitive, well, we started off with Luminex and measured a large number of host analytes. When we narrowed it down, we had this very primitive lateral flow test, and now we are actually busy with a cartridge, and we are right before a lockdown design for a test that we are wanting to present to the WHO for potential endorsement, at least for their advanced recommendations of what is needed next to actually consider endorsement as a triage test. 90% sensitivity and 74% specificity. Not too shabby and very similar to the Gene Expert MTB host response test. Very similar performance characteristics. So we have 2 tests that could be used as triage tests, but more about that later. So the new kid on the block is actually not that new. It's been around for decades, but now people are becoming more interested in this topic. Is subclinical TB, asymptomatic people who are going to develop or not going to develop TB. And there was recently a consensus meeting in Cape Town with a whole lot of people from all over the world participating, and a suggestion was made to divide the TB infection and disease spectra into 5 different classes. The first one is no TB infection. And then, so there's no macroscopic pathology present, like on X-ray. Symptoms are absent, microbiology is absent, but these people can be infected, and we can only diagnose that by immune tests like the tuberculin skin test or equivalents, or the interferon gamma release assays. So they have MTB infection, but nothing else. Then the first stage of subclinical TB would be the non-infectious stage, where there is some pathological evidence of disease, and usually this is on X-ray or on CT scan or on PET-CT, where there must be minimal and very restricted signs of lung abnormalities. Symptoms are, again, totally absent, microbiology negative. And this was previously called closed TB or minimal TB. So I'll talk more about the significance of this in a minute. And then there's infectious subclinical TB, where there is, again, X-ray presence. So it can also be absent. No symptoms again, but there is at least intermittent positivity of midget culture or expert testing. And strikingly, about half of prevalent TB cases, when you do community screening with expert, for instance, half of all TB cases are this infectious subclinical TB. So this is not something we can easily ignore. Stage 4 then is extrapulmonary TB. I'm not going to talk about that. And then lastly, clinical TB. So 50%, let's say 50% on average, of prevalent TB is subclinical asymptomatic. Now that is a problem. How do we find those? Except with community-wide screening, because these people will not come to you. Like I said, on imaging, chest X-ray change will be minimal or subtle. And we have seen in our data, and Emily Wong in her data, 4% of the general population in high prevalence settings have some form of subclinical TB, 4%. 1% of our population has active TB. And in close contacts, like household contacts, 10% of people have one other form of subclinical TB. And the more you look, the more you find. With CT scanning, 25% of close contacts have signs on X-ray that suggest subclinical TB. And microbiology, like I said, is scanty, low positive on gene expert. We would not regard somebody with a high expert as subclinical TB. Well, we would. We still have to call it subclinical. We would treat that. But the rest, asymptomatic, scanty microbiology, and scanty X-ray, what do we do then? In high prevalence settings, about 1% of the population develops TB each year. In our country, it's about 600,000 people each year out of 6 million. In close contacts, it is 2 to 3% of close contacts develop TB each year. And if you look at subclinical TB, it's 10% that develop TB in 1 year and 25% over 3 years. Okay, 10%. So 90% don't develop TB. But 10% is a significant number, and we can't ignore that 10%. Particularly because we now know that more than half of TB transmission happens from subclinical TB. And the reason is probably the length of exposure that contacts have with such a case, because they are asymptomatic. They don't have a high bacterial load, but they probably do transmit every now and again over a prolonged period. So more than half of transmission seems to come from subclinical TB. So we can't ignore subclinical TB. Here's some images, PET-CT images from our site. Here you see a typical active TB case. Here you see some of Hanif Esmail and Robert Wilkinson's HIV-infected individuals with subclinical TB. Before and after INH treatment, you see this one here, before treatment, after INH treatment for 6 months, it's gone. Here's another one, it's not that obvious, and this is what we often see, is this type of lesion. And you can see it diminishing after treatment. This one maybe did not really change much, it's a bit more dense and a bit smaller. And here's another one that responded to full TB treatment. So here's an example of a PET-CT inactive. So there's a lesion, but it doesn't show high uptake and it doesn't have the characteristic imaging criteria for active TB. It's not cavitation, it's just a nodule. So this person has a 1.2% chance to develop active TB over 5 years. But then we have a more typical form here. You have PET-CT active. You see here the 3 in bud changes. So this was probably a spread via the bronchial veins. So a segment here that is supplied by a pulmonary vein that disseminated these multiple granulomas throughout the segment of the lung. And this type of person can stay asymptomatic until they convert to active TB, and this is in 35% of cases. You might not see this on a normal X-ray. Okay, and then to show how imaging is not enough, here you have a person with active TB, symptomatic, hardly anything to be seen on X-ray except up here. This person is microbiologically positive, has symptoms, and has active TB. So it's difficult. This is the noise that Bruno was probably referring to. The other phenotype that I am very interested in is end of treatment. Do the majority of treated people revert back to subclinical TB? And here you see 2 examples. The person at baseline after 1 month of treatment, actually extension of the inflammatory changes. And here at month 6, a lot less inflammation. Remember, PET-CT only indicates increased glucose uptake, radioactive glucose uptake. It is not specific for TB, it just shows inflammation. And this person then subsequently relapsed after this person was clinically cured after 6 months of, or actually this one was after 4 months of treatment. This person met certain treatment cessation criteria, was stopped on treatment after 4 months, but then relapsed. And here's another one. This one is more obvious. After 6 months, there is a hot lesion. And although this person was microbiologically cured and asymptomatic, developed relapse in the other lung following cessation of treatment. So I want to propose that end of treatment is a great place to look for subclinical TB if you want to do research on that. This is Novel Chegu, who some of you know. He has done a lot of our biomarker work and amongst others, working on extrapulmonary TB, TB meningitis, which is a dreadful disease to get. 2 to 5% of children who get TB have TB meningitis, 20% die, and up to 50% have permanent severe neurological sequelae. Diagnosis is difficult because it's clinically not distinct from viral meningitis or just fever, high fever. The CSF cells and the biochemistry lack sensitivity. Often the cultures are negative. The smears are mostly negative. Expert is quick, but only 50–60% sensitive. And it is really very important to diagnose these kids quickly, start them on treatment, and hope to prevent some of the long-term sequelae. The WHO, anybody from WHO here? They dreamt up these target product profiles, 80% sensitivity, 98% sensitivity. Nothing we have, not a single test or a combination of tests, except follow-up, actually gets close to that type of performance. I don't know why they come up with these. Because now somebody does a biomarker test and they find 70% sensitivity, which is better than anything we have, and they dump it because it doesn't meet these aspirational TPPs. So Novel looked at 67 host markers in CSF, and he had 87 kids, of whom 44% had TBM, 2 had viral, 6 had bacterial meningitis, and 41 others had febrile illness without meningitis. And you don't know until you've done the lumbar puncture. So this table is a terrible slide, right? But what it shows is that the sensitivity and specificity of 3 to 4 markers in the CSF actually perform pretty good. Granted, there's a lot of noise and there's a small study and it needs repetition, but 84% sensitivity and almost 90% specificity would be a game changer in this type of setting. But what we need is sufficient numbers of these kids on the same treatment protocols and the same investigational protocols to confirm such performance, and to either move on and develop these into point-of-care tests or patient near tests, or to close the book on this type of approach of host-based biomarkers. Novel also looked at extrapulmonary spinal TB, got a 100% sensitive and specific assay. Again, small studies, but where you find enough spinal TB cases who are worked up properly, who have the right samples taken to confirm this type of performance. So he has 5 markers with a great predictive performance for spinal TB. Spinal TB is very difficult to diagnose. It often looks exactly what Jerry Ellen and I experience, due to being on this planet for a long time and walking upright. Back pain. It is difficult to diagnose. And eventually it requires either PET-CT scanning or a biopsy. So if we can screen out the people who need that biopsy and the CT scan with such a test, that would be a great advantage for biomarkers. Just briefly, our treatment response study, we used PET-CT and gene expert CT values together with David Lalande to do a treatment shortening study on HIV uninfected people in China and South Africa. Cut a long story short, we just see what happens in the next slide. No. So we stratified people at baseline into a conventional arm if they exceeded a certain extent of disease score, or randomized them into a shorter or a standard treatment arm if they had a lower extent of disease score. The shorter treatment arm led to too many relapses. The study was stopped. And we have, and by the way, report 2 contributed to the sample collection at the final time point for these patients. And we are currently looking at a large RNA-seq experiment on those people, on the relapses, plus controls to find transcriptomic signatures for high risk for poor TB treatment outcome. Donald Simon and Candace Snyders looked at Luminex on 50 markers on a subset of the patients. And surprisingly, from 50 markers, only 3 showed any difference between the relapse and cured group at any time point. TNF receptor 1 at baseline, TNF beta at the end of treatment, and then IL-22 between the two groups at week 16 and week 24. That's a bit disappointing. Donald went ahead and did all kinds of modeling exercises and came up with some signatures that were reasonable. 86% sensitivity and 70% specificity, but 7 host markers plus VMI plus previous smoking plus previous TB, maybe a bit too large a signature to be really of clinical use. So we are working on the transcriptomics now, and we will be doing proteomics and metabolomics as well, and try to find a small as possible signature that could be used clinically or in clinical trials to show us when is treatment enough and who has a high risk for a poor outcome. So this is not the end of that story, that's ongoing. So some reflections on host biomarker test. So the whole expert MTB RIF rollout has really made a biomarker test based on the same instrument unnecessary. Why would I do a host response free transcript test on blood if I could do a sputum test, a definitive sputum test that leads immediately to treatment? So this is not a test. This Cepheid expert MTB host response test is not really an option for pulmonary TB in adults. It might be for special circumstances. For instance, in the subclinical TBs, close contacts, in high-risk populations, where the microbiology is still negative, but we don't know yet. We haven't tested that in that cohort yet. Treatment response, as soon as the bugs go away, can we use this? We are busy with, on the FEN project, we are busy looking at the expression of those 3 genes, and we're following people up for relapse. Children may be a special group where such biomarkers would help. They are often sputum negative. It's difficult to get sputum. And then there are, of course, in Africa and Asia and South America, lots of population who don't have access to immediate expert MTB RIF assays. And this is where we believe that our protein test, which is now in cassette form and can be measured on a handheld reader, might come into its own right, where a healthcare worker on a bicycle could log this around with him and test symptomatic people. Extrapulmonary TB, again, that needs a host-based biomarker, and any form of possible ciliary disease, maybe in HIV. Okay, that is it, and I I'm just the traveling salesman. There are a lot of collaborators all over the world, a lot of funders, including Report, who have contributed to this world. And Bruno, we are trying to find markers that can shout through the noise. Thank you.
Thank you.
It's not something you brought up in your talk, but I'm seeing papers these days claiming to find circulating TB DNA
Yes.
in blood.
Yes.
And I don't know, they take different approaches. One of them is a phage-based assay. And actually they've approached Report India and they'd like us to work with them. But I find the results surprising. Have you done much work looking for circulating bacteria? We did that with Expert.
Circulating bacteria or their DNA?
Well, we looked at DNA by Expert.
Yeah.
And maybe 10 or 20%.
Yeah.
About the same number that have positive blood cultures.
Yeah.
But we don't see 70 or 80%, and we don't see positives in LTBI. I just wondered what your perspective for.
I mean, we know that TB DNA lasts for a long time. You can find TB DNA in about 30% of sputa from cured TB patients. So 2 years after cure, you might still find positive DNA. Even RNA, we're not sure how long that is actually available, measurable once the bugs are dead. We have shown in bronchoalveolar lavage after curative treatment, almost all our patients have MTB RNA. MTB RNA or any RNA is supposed to be degraded immediately, but maybe it's different in TB with its thick cell wall. So I don't know. You would also have seen these CD34 positive cells, these stem cells that have TB in them, culturable MTB. And so I think, like Bruno also said, we actually know very little about what TB does in us, to us. I think eventually a pathogen-based test will have to be found that deals with this. I'm just trying to find something in the interim until we have that test that helps us to stratify people into risk groups.
I'm wondering what's the status of Bill's this phage test where the phage infects the bug and it packs up, right?
Yeah.
So has there been any?
This company Actiphage has a variety of that. This phage-based diagnostics.
Because there you're looking at
also phage-based therapy.
Yes.
So that was done in a small number of patients who had immunological deficits and had recurrent infections, often with NTMs. It did not sound ready for prime time to me. It was quite difficult to find the right phage for each individual patient. I think it sounds great, but I haven't seen data that convinces me that we are getting close.
Because that would bypass, it would detect live bug. So.
Yes.
Do you have any insight into the use of extracellular vesicles to detect bacterial products?
Yeah, so Karen Dobos at CSU is doing that, and it sounds really promising. She claims that she can see mycobacterial proteins in even latently infected people, and we are actively working with them, also on this PREDICT study, to investigate that further. But the extracellular vesicles are certainly a promising avenue, I think.
Gerhard, this is Mary. May I ask a question, Paddy?
Yeah, sure.
Yeah, I was actually going to ask a very similar question to what Marcella just asked about the extracellular vesicles, because if you say that you find that people find bacterial RNA, for example, in circulation, it could be protected by the vesicles, right? because
Yes.
if there is free floating RNA, it's unlikely that, I mean, sorry, not it's unlikely. It's unlikely to be found. It would not be stable enough. But
Precisely.
it could be protected by vesicles. By the same token, these vesicles could also, contain markers of cell death or cell damage or cell unhappiness.
Yes, absolutely. Agree. I mean, that brings me to these differentially culturable bacteria, which is also a difficult field. And why it is difficult is because I don't think the assay is done correctly. So the assay is done by adding RPFs, basically culture supernatant, to dilutions of MTB, or to dilutions of sputum. And when the wells turn fluffy, you say, well, this is TB. Well, it's not. Caroline Beltran in our group has looked at that. You can't use the fluffiness of the wells to equate that to TB, because there are a lot of other things in sputum that also grow. You can't do PCR for confirmation because the material that you add is culture supernatant. So what you have to do is you have to subculture that fluffy well and pick colonies, if they grow, and confirm that it's actually MTB. We've done that and we found these differentially culturable bacteria in only 10% of patients at the end of curative treatment. And about half of those relapsed. The other half did not. So I don't think that assay is done properly, but it does suggest, together with the ongoing PET-CT inflammation in the chest, that non-sterilizing cure happens. And of course, non-sterilizing cure happens, otherwise you wouldn't have relapse. And we also don't see that everybody with those differentially culturable bacteria develop relapse because we have an immune system. If we don't have a good immune system, like in diabetes or in HIV, relapse rates are indeed higher. So why would that be? The bacteria should still be susceptible to MTB. So I think it's an interplay between the antibiotics and the immune system that keeps relapse in check. I think there's non-sterilizing cure in at least 10% of people. But we have to find a way to find those 10%. And then we need to look earlier during the treatment period, like 3 months, 4 months. If they don't have that magic test, we can stop treatment. But we don't have that magic test yet. And I think Report has a wonderful opportunity to contribute to such investigations, because there are very few cohorts in the world with adequate numbers of relapses that are coupled to sufficient clinical data and samples. I think we are currently working on the biggest collection of relapse samples, and that is 40 relapses. It's pathetic. But that's the biggest there is in the world. And I think in Report, if you look at 1, 2, and 3, Report 1, 2, 3, there might be sufficient relapses, confirmed relapses, that we can use as a validation if we find anything of interest in our current work.
Can I ask a follow-up question?
Of course.
Why do you need to grow this bacteria? I mean, there are methods and platforms that they can detect, at least they claim they can detect single molecules.
Like LAM.
Yeah, no, I mean, there are these, CMOA platforms, etc.
But does a single molecule tell us whether they are live bacteria?
True. Yes. No, of course not. No.
Microbiologists are just lagging behind.
David's not here.
Yeah, but yes, true. Yes, I'm also a microbiologist by training, but so I sympathize with the idea, but it may be a tall order to grow this bacteria, right? They may not, like you said, they may not grow or grow differentially or grow. So there could be phenotypic differences that make it super hard to grow. So maybe finding them is
They become cell wall deficient, unstainable, or unculturable
Yeah, exactly.
until conditions are ripe for them to switch their gene expression profile. So I think proteins, MTB proteins, would be good, but I'm not sure how long those will last in circulation if DNA and RNA hangs around for years.
Well, I don't know either, of course, but they wouldn't last forever. I mean, they would be cleared, right? They would be clear. I mean, if there are no bacteria producing them, if the bacteria are dead, they wouldn't last. I mean, I don't know how long they would last. It's testable. It wouldn't last for.
Well, I
They
would have
would be
expected them
cleared.
to last shorter than RNA. RNA is present months after curative treatment. There's one in the audience.
Yeah, so I have a question on the subclinical TB definition. How stable is this population? I mean, it looks like the definition of that subclinical is so ambiguous and not really well defined. Is that subclinical is like a primordial for an active TB, or are they just to sustain like a trough for a long time?
In 10%, it is a pre-clinical disease state. 90%, it is not. So that is the whole problem, because you treat the whole lot, 90% unnecessary. By the way, asymptomatic people don't like taking drugs. So it is in some people, it is a pre-stage to clinical TB, but not in all. That, I would say, as an immunologist, is due to the immune system.
And how would you discriminate between those two? They both come under subclinical TB.
Yes. So the one has a positive culture, and the other, or gene expert, that's the infectious subclinical TB, and the other one only has imaging changes that can be associated with TB. And there are so many other things that can have a similar picture on CT or X-ray, so that it makes it difficult again.
So it's basically a matter of time.
Subclinical and clinical is.
So it's basically a matter of time when you diagnose a person, whether it's a subclinical or clinical, right?
Yes.
It's not clear whether even subclinical infections will all progress.
No. Most don't progress. Most don't progress. The vast majority don't
Yeah.
progress.
Yeah.
But can we ignore the 10% that do
Yeah.
progress? And there are studies now showing that more than half of transmissions happen due to subclinical TB. And that is striking. So we can't ignore subclinical TB any longer, even though the Gates Foundation does.
Gotcha.
There's another entity that's not talked about much, but it seems to be rearing its head in some of the studies that Padmini and Linda have been doing, and that would be culture negative tuberculosis. And this also might be a reason to look at people's respiratory symptoms with some sort of biomarker.
Yes.
Do you have any comments about, what are your thoughts about how frequently that is an issue?
Yeah, it is difficult to find those in self-reporting patients. In a pulmonology clinic, you'll find them because there will be an X-ray abnormality, cultures will be negative. You'll eventually do a bronchoscopy that might be negative, eventually do biopsy that will be positive, or the person will progress and become culture positive. We are cowards. We focus on easy to get in carbs.
That's one.
Yeah, or it's so prevalent, right, that you can't, right? It's hard to address. Can I ask you a question too? Okay, so this is something I've been thinking about a long time. This might just be a crazy idea. So my background originally came from cancer. And there are certain cancers that that have kind of gone down, like for example, breast cancer or something. And the classifications went from clinical classifications to now molecular classification. So there's specific molecular classifications in that space based on gene expression or something like that. And that has actually led to different treatment approaches based on molecular classifications. And I think in TB, a lot of times the clinical classifications are they tend to dominate everything. So any thoughts on should we be making a transition to some sort of molecular-based classification of TB and then have that drive treatment?
So would that then be like bacterial molecular classification?
Yeah, or gene expression or some, what are your thoughts?
So what we are doing now is looking at drug acceptability, non-resistance, and extending our testing to multiple drugs, those where it is possible. Then we have the problem of phenotypic versus genotypic resistance. And we know that there are many new mutations that we will not catch with our targeted gene expression approaches or gene approaches. That field is evolving fast. And there will be in silico, as you in your field, there will be in silico predictions of certain mutations that will lead to drug resistance. But I think there are so many possibilities there that it will take a while to really catch up. Phenotypic takes much too long, is much too expensive. By the time we get a response, well, firstly, by the time we request the phenotypic drug resistance test, the patient is half gone. And by the time we get the results, it might actually be too late. So I think genotypic assessment is going to be the way to go. But it will require probably some machine learning to really predict which mutations actually will lead to resistance. Do you agree with that?
Yeah, I hope so, because we're kind of working
on that.
We're working on that, so hopefully it works.
Thank you so much, Gerhard.
Bye, Gerhard.
For joining everybody else.
No, nice seeing you even.